Hi, my name is Jessica Spires, and I'll be talking to you today about the next frontier in PBPK modeling. We'll be discussing the delivery routes that move beyond the oral administration and how we can use PBPK modeling through our GastroPlus software platform to understand the effects of formulation characteristics on the drug absorption from these routes and on the local and systemic drug concentrations. Generic drug delivery for locally acting drug products is, of course, a very fast-growing field of interest, in particular, with a lot of interest from the FDA and other regulatory agencies. But when we're looking at these locally acting products, the traditional bioequivalence approaches may not be relevant or applicable. Little or none of the drug may be reaching the systemic circulation, so there may not be any plasma concentration to look at. Or even if it does, it may not really reflect the concentration profiles in the local tissues that we're most interested in. Bioequivalence for these locally acting products can be assessed through in vitro testing and/or in vivo testing, depending on the exact situation. But of course, the in vivo testing in particular can be very difficult or expensive, and especially when trying to assess local concentration or effect data. So this is where modeling and simulation can play a role in supporting the development of these products. The composition of the formulations of these locally acting drug products can strongly affect the absorption of the drug itself, of course. In particular, formulation attributes like the particle size, the solubility or the diffusivity of the drug in the formulation, and the formulation viscosity can all be key factors that are affecting the local absorption. However, understanding their importance really depends heavily on the local physiology of the route of administration as well as the nature of the formulation. When we have a good understanding of these physiological mechanisms of absorption in the area of interest, as well as a good understanding of the characteristics of the formulation, we can use in silico physiologically based modeling to simulate the processes by which these formulation attributes are influencing the absorption. If we're able to predict the effects of these formulation attributes on absorption and local concentration, then of course we can use this to inform the development of the generic formulations while reducing the need for animal and human testing. One of the best ways to perform this sort of in silico modeling is by using the GastroPlus software platform, which of course is made by Simulations Plus. GastroPlus is the premier platform for PBPK modeling and modeling of the orally administered drugs. So we're very well known for our simulations of absorption, biopharmaceutics, pharmacokinetics, and pharmacodynamics of drugs or chemicals in humans and animals. Okay. It's also an excellent platform to model the absorption from these complex dosage routes as we move beyond the intravenous and the oral. So currently we include modeling of delivery to the oral cavity, to the eye, to the nose and lung, and to the skin, as well as the subcutaneous, intramuscular, and intra-articular injections. Today, we will be focused on modeling three of these pathways: the ocular, the dermal, and the intra-articular. In the case of the ocular and the dermal routes, we have ongoing collaborative grants with the FDA to expand and improve our modeling capabilities, particularly in the areas of the formulation development. I'd also like to talk about the intra-articular route because it's the newest route of administration that we've added. So to give you an idea of how you can use these models and use similar strategies as you model the additional dosage routes. So we'll start off talking about the ocular delivery through the OCAT model. The eye is, of course, a very complex organ, and so our ocular model is similarly complex. We divide the eye into 13 compartments, and we include all of the different transport processes between them, and I'll go into a little more detail about those later. As you can see, we also split several of the ocular tissues into multiple compartments, and in some cases this is due to physiological differences, for example, dividing the cornea into corneal epithelium and stroma. In other cases it may be, for example, to better model diffusive processes, for example, to model slow diffusion within the vitreous humor. This slide shows all of the ocular dosage forms that we include in our OCAT model. For the topical administration, we include topical solutions, suspensions, and ointments, and all of these would be applied to the precorneal film. We also include the intravitreal injections, particularly solutions, suspensions, and controlled release implants. And finally, we also include the subconjunctival implants as a dosage route. We also include default ocular physiologies for the eye. As of the latest version of GastroPlus, version 9.8.2, we've included an updated model for the human eye, as well as rabbit eye models for two specific breeds. Those being the pigmented Dutch-belted rabbit and the albino New Zealand rabbit. In addition to those, we also have a more general rabbit eye physiology and also a monkey eye physiology. In addition to these, we do allow users also to create and save their own custom physiologies. So in some cases that may be to model another species or maybe to model a pathophysiological condition. We have the same capability with all of our complex dosage routes. We've done a series of grants and contracts with the FDA to improve the OCAT model. Currently, we're in the middle of a 3-year funded collaborative project with the FDA Office of Generic Drugs to expand the OCAT model to predict human ocular PK and PD for ophthalmic formulations, including the more complex formulations. So you can expect more improvements and features to be coming to the OCAT model over the next few years. We've also published several manuscripts describing the use of the OCAT model. You can see some of the citations here, and today I'm going to focus on the one that's outlined in red. This was a case study where we applied the OCAT model to dexamethasone topical suspensions that were applied to the rabbit eye. As you'll see in the next few slides, we were able to use the OCAT model to understand the impact of the formulation properties on the local ocular concentrations. I'll mention as well, the lead author on this one is Maxime Lemerdy, he was at the FDA when this manuscript was published, but since then I'm happy to say that he's joined us here at Simulations Plus. Dexamethasone is a lipophilic glucocorticoid. It can be used for a few different things, but in the context of the eye, it's usually topically applied to treat local eye pain following ocular surgery. In the paper that I'll be discussing, we use the OCAT model to characterize the ophthalmic dexamethasone suspensions and explore the relationship between the formulation characteristics and the physiological clearance in the rabbit eye. The FDA was able to provide measured concentration time profiles of dexamethasone that they had measured, not only in the plasma, but also locally in the cornea, the conjunctiva, and the aqueous humor. We knew what the formulation was, so we were able to input its characteristics and use them in the model. Then we used this data to build a baseline model of dexamethasone in the rabbit eye. We fit the permeabilities in the conjunctiva, aqueous humor, and iris ciliary body with other permeability values taken from the literature or predicted from the compound structure. As you can see from the plot, points are the experimental data, the lines are our simulated outputs, and you can see that we were able to match the plasma concentration and all of the local concentrations. So we were happy with this as a baseline model. We were able to verify the model further by modeling additional dexamethasone suspension formulations that were applied to the rabbit eye with different strengths, viscosity, and particle size. We modeled the effective viscosity changes by adjusting the drainage rate from the precorneal film. With this viscosity adjustment, which was according to the different viscosities of the formulations, we were able to predict the effect on the aqueous humor Cmax and AUC. We also had cornea concentration time profiles for suspensions with different mean particle sizes. And as you can see, the model was also able to predict the effect of these different mean particle sizes on the concentration time profiles. So with this further verification of the model, we were very happy. So we went further and we started using the model to predict the effects of formulation changes. To do this, we performed parameter sensitivity analysis to understand the sensitivity of the local ocular concentrations and the plasma concentration to the changes in the particle size and viscosity, with a particular focus on the cornea and the aqueous humor. We found that there was high sensitivity to the viscosity, but a somewhat lower sensitivity to the particle size. as well as some other conclusions that you can look at in the manuscript. Finally, we explored some potential hypothetical formulations. These are cases where the hypothetical formulations had similar mean particle density and similar D10, but different D90 values. So we wanted to see, would this range of variation be acceptable for ophthalmic formulation? Are they similar enough? What we found was that within this range, the simulation results showed little difference in the aqueous humors Cmax and AUC. So as a result, that would suggest that this range of variation is acceptable for an ophthalmic formulation. Next, we'll talk about the transdermal and the topical delivery through the TCAT model. We can model absorption into the skin and local skin concentrations using the transdermal compartmental absorption and transit model, the TCAT model. I should say, despite the name, it can be used for both transdermal and topical formulations. The skin is the same, the only difference is the intent of the user. In this model, we divide the skin into the epidermis, distinguishing the stratum corneum and the viable epidermis, and also the dermis beneath it, which is going to be the vascularized part of the skin. We also include the subcutaneous fat as part of the TCAT model. In addition to including these kind of major parts of the skin, as you can see, we also include the hair follicle unit. The hair itself is split into the hair core and a hair lipid layer surrounding that core. And within the skin, we include the sebum that's surrounding the hair, which can be an important alternate absorption route for some compounds. The dosage forms that can be used with the TCAT model are shown here. We include solutions, suspensions, ointments, pastes, and then as well as the emulsion dosage forms, so lotions, gels, and creams. We also include the reservoir patch formulation, and this would be the case where the patch controls the release into the skin. For the TCAT model, we have a 2-year funded collaborative project with the FDA Office of Generic Drugs. For this grant, we're improving the TCAT model further by enhancing our modeling of formulation mechanisms as well as our modeling of the skin physiology. So just as with the OCAT model, you can expect even more expansions and improvements to come. Now I will discuss modeling of topical formulations with the TCAT model through a case study that we developed as part of a previous grant with the FDA. Here I'm summarizing work that we presented previously at our MIDD Plus conference. It was presented by Dr. Bill Van Osdal, my colleague, and he and our colleague Yasmina Novakovic did a lot of the work on this project. So clobetasol 17 propionate is a glucocorticoid that's used topically to treat inflammatory skin conditions such as psoriasis. When it's applied in formulations such as oil and water microemulsions, the dispersed phase droplets can act as a reservoir for clobetasol to maintain its concentration in the aqueous continuous phase. We decided to model this process, specifically using experimental data from applied Dermavate cream. In this case, we actually had local concentration data in the skin that was collected in vivo in the dermis. And this was through a technique that's called dermal open flow microperfusion. And I recommend reading the Bode-Lins paper if you'd like to know more about this. Okay. And we also knew the probe depth at which the concentrations were taken. Here on the left plot, the points that you see are the experimental DOFM data. And the lines that you see are the simulated concentrations from our baseline model of clobetasol in the dermis, right around the depth of the probe. Okay. And as you can see, we found some pretty good correspondence between these. On the right, you can see the AUC in the dermis at different skin depths. The blue circles are the experimentally measured values from the Bode-Lins paper, and the red squares that you can see here are simulated dermis AUCs at different depths. As you can see, we pass through the range of the observed values at the different probe depths. With the verification of the baseline model, we then move to explore the sensitivity of the local dermis concentration to formulation parameters. In particular, 2 formulation attributes we were interested in were the effect of the dispersed phase solubility, which we adjusted through the dispersed phase water partition coefficient in the model, and the volume fraction of the dispersed phase. We found that as that dispersed phase solubility or partition coefficient was increased, more clobetasol partitioned into the dispersed phase, lowering clobetasol concentrations in the continuous phase. In turn, this reduced the driving force for clobetasol to partition into the stratum corneum, which reduced concentrations locally in the dermis as well. We saw a similar process taking place with increases in the dispersed phase volume fraction, although as you can see, the effect was less pronounced. Finally, we considered the combined effect of both of these formulation attributes via a coupled PSA. We simulated a virtual factorial experiment design, and we were able to derive a response surface for the effect of these parameters on the 24-hour AUC in the dermis and support predictions and interpolated values. And you can see how this kind of response surface could then be used to inform future formulation developments. Finally, I'd like to talk a little bit about the intra-articular delivery route in GastroPlus. Here you can see a diagram of version 1 of the ICAT model, which is in the current GastroPlus release. The intra-articular doses are going to be injected into the synovial fluid. From there, they can diffuse into the cartilage, or they can diffuse through the synovial membrane and into the systemic circulation. And as you can see, all of these arrows are double-sided, so compound can diffuse back and forth in every direction. The ICAT model includes default physiologies for the human, dog, rat, monkey, and mouse. The dosage forms that we currently include would be the solutions, the suspension injections, and also controlled release into the synovial fluid. I won't go into too much detail about the input parameters, but one important aspect that we need to think about when we're modeling the intra-articular injections is correctly modeling the diffusion processes. Currently, we allow the user to estimate the diffusion coefficients in 3 different ways. Using the Stokes-Einstein equation, using a diffusion estimate in cellular tissues that we previously developed for the oral cavity model based on log P. And then the third option is to simply use the aqueous diffusion coefficient to model diffusivity in the compartments. The case study that I'll talk about here is a compound whose name I will not even try to pronounce, but we can call it TCA. TCA is a corticosteroid. It's often injected intra-articularly to treat joint problems such as pain due to osteoarthritis. We wanted to see how well we could model the TCA intra-articular suspension injections. And to do this, we actually ended up pooling data from 2 different injections with the same dose. For the first 24 hours, we used data from a Kinalog injection, and all of the data after that came from a Volon A injection. Those concentration time profiles that I showed previously came from the plasma. So the first thing that we did was actually to build a PBPK model of TCA so that we could describe its distribution and elimination in the systemic circulation. That way, when we go to model our intra-articular injections specifically, if we run into any trouble, we know that it's a problem with the absorption from the injection site and that our modeling of the distribution and the elimination from the systemic circulation is correct. As you can see, we were able to build that PBPK model, and it's able to describe the PK of TCA for IV bolus injections, as well as an oral tablet. The next step was to model the intra-articular suspension injections. And for this, we used the particle size distribution of the Kinalog injection. For the initial model, we assumed a diffusivity in water for the synovial fluid, and the diffusivity in the tissue based on the oral cavity model for the tissue diffusivity. So what we found in our results, and you can see here the blue squares are the experimental data, and the blue line is the predicted, is the simulated plasma concentration. You can see that initially we saw about the correct rate of compound entering the systemic circulation, but then the synovial fluid became saturated. Okay, this caused a plateau in the simulation of the plasma concentration that was not present in the experimental data. To calibrate the model and get closer to the observed PK, we made some different assumptions on the diffusivity. In particular, we used the Stokes-Einstein correction for the diffusion coefficient in the synovial fluid, and then we assumed an aqueous diffusion coefficient for the other tissues. Using these settings, as you can see, we were able to get a very nice match to the experimental data. So, end of story, right? Maybe not. We also considered an alternate explanation for the profiles that we observed. From the literature, we found that commercial TCA injections can agglomerate over time while they're sitting in saline, even if they're diluted. So we want to explore whether this agglomeration process could be causing the results that we see in vivo. So to model this, we adjusted the particle size distribution until the model results matched the experimental data. We were able to match it, as you can see here. However, in order to get a match, it required about 50% of the total undissolved drug to be agglomerated into the equivalent of 2,000 micron particles. We said, even if there is agglomeration happening at the injection site, this may be a little unrealistic. And the results that we get also don't match quite as closely to the experimental data as they did when we used the Stokes-Einstein equation instead. So this is a case study that's still in progress. We have two hypotheses for the mechanism leading to the PK that we see in vivo, but it's a little difficult for us to distinguish between them currently with the data that we have. Our hope is that as we continue to model more formulations and more compounds, and we'll be able to distinguish between these processes and give more guidance to modelers. But in the meantime, we have released the ICAT model so that users are able to use it with their own data, and perhaps distinguish these mechanisms for themselves. So to summarize, we've spoken about three very different dosage routes, and hopefully you can see some of the common factors that should be considered as we move beyond simple oral administration. PBPK modeling through software platforms such as GastroPlus can be used to build and validate models of locally acting drug products across a wide range of routes of administration. These models are able to simulate and predict the effects of formulation attributes when those attributes are well understood and the physiology of the system is well understood. They can also be used to identify which formulation attributes are most critical in determining local absorption and local and systemic concentrations. And putting it all together, it means that a verified model can be used to predict the effects of hypothetical formulation changes on local and systemic PK. Thank you very much.